We have Dr. Moser, Uveal Melanoma medical oncologist here in Scottsdale, and our hosting doctor from Honor Health Research Institute. Dr. Moser has a special focus on uveal melanoma and has an active translational research program focused on a better understanding of this rare tumor. He is actively developing new clinical trials and treatment options for patients with this disease. We have Peaches Ulrich. Dr. Ulrich has extensive background in molecular and cellular biology, immunology, and experience as a molecular science liaison. Throughout her career, she's developed a deep understanding of the intricate workings of molecular and cellular systems with a specific focus on immunological processes. Dr. Moser and Dr. Ulrich, welcome to the stage.
Thank you.
Well, that was a long introduction, but it all basically means that I'm a little pocket nerd. I studied the immune system for a very long time in grad school, and now I work with a company, a disclaimer, called caregivers. But essentially we do somatic testing, like a lot of other tests that I'm sure you've heard of. And so I was invited by Dr. Moser today to talk just in general about somatic testing. What is it? Why do we do it? How do we do it? So I had slides, but we're going to kind of wing it today, and we can make this a discussion based and ask your questions very informally.
I think we all know about the genetic testing of primary tumors through Castle. So I'll just start by giving kind of a high-level overview about the different types of genetic testing, and then we can talk about the somatic test and metastatic tumors, if that's okay.
Absolutely, yeah.
So, as most of us are aware, when you have a tumor in the eye, we can do genetic testing on that tumor with a biopsy through Castle. And that's really a focused genetic test where they're looking at certain types of RNA or DNA to try to predict how aggressive the tumor is going to be. And so when you have genetic tests, you can always test the person or the tumor. So that's one of the tests where we test the tumor, and we're doing it in a focused way to try to predict something. We then have where you can test the person. So I think this is most commonly seen in breast cancer, right? We know that there's different mutations you can be born with, maybe in BRCA, where we can test your DNA to see if you are at higher risk for getting cancer. And then once patients develop metastatic cancer that's spread and we can't cut it out, we can actually test the tumor's genetics, not for a specific test or prognosis, but kind of as a shotgun approach and say, I call it a fishing expedition. We're basically testing the cancer to say what mutations or changes in the DNA or genetics has this tumor developed, either to better understand it or try to find a way that we can exploit that and use it to guide treatment. And that's really what this talk is focused on, that kind of fishing expedition where we test everything to try to figure out, is there a better treatment option or a personalized treatment option based on that genetic testing? And that's really what Peaches specializes in her career.
Thank you. I'm big on analogies and metaphors. I'm a visual person, so apologies for not having the slides. But I think about somatic testing as essentially looking at a house. So as an individual, we are organisms. We're made up of different organs, and our organs are made up of different cells. In the same way, we have, if we think about a house is made up of different rooms, and every room has different features that make it a characteristic of that room. So if I walk into a room and there's a toilet, I think, hey, that's a bathroom. If I see an oven and a stove, I'm thinking the kitchen, a bed, a bedroom. And our bodies work the same way. And just like you can have a house, it could be built in the 1960s or it could have been built yesterday. There's this master blueprint for houses. And that's how our DNA is. Our DNA is the master blueprint of our bodies, whether it's normal, healthy tissue. And sometimes what happens is those blueprints get messed up a little bit, and that leads to our cancer. And so just like a house, it can be made, and you walk in the door and there's a squeaky door. Hey, DW40, kind of an easy fix for it. And that's what we're looking at in cancer, in somatic testing. What are those differences from the healthy tissue, from the manual blueprint, and how can we fix it? And so with a lot of traditional medicine, our chemo, radiation, surgery, those are all part of the toolbox that are still used today. They're great. But with somatic testing, it doesn't only tell you that there is a problem, it tells you more about what the problem is to inform treatment. So if you walk in your house and there's water all over the floor, obviously there's a problem, but is it due to the faucet being left on, a pipe broke, or maybe the city did some construction down the street? Knowing which of those problems it is the source is important, because if you know the source of the problem, it gives you better informed on the treatment, on how the oncologists like Dr. Moser are going to then treat your cancer. And so somatic testing is just that other side of traditional medicine that informs you more about what is different in your tumor than the healthy tissue, and how can that information inform treatment. And so there's a lot of companies out there, just like Caris, Tempest, Foundation, lots that do genetic testing, but all of them are essentially looking for three major things: changes in your DNA, in your RNA, and protein. All these different things that can be changed in the tumor tissue versus your healthy tissue.
Great. So I think that's a really good overview of kind of what this testing is. So any questions on kind of the basics of what is somatic testing, which is also tumor testing, right? Somatic just means tumor testing. Any questions about that before we kind of go in deeper and start talking about the different types of tests that are out there? When we talk about somatic testing, Peaches, that just means we're testing the tumor. But there's obviously different platforms out there. There's ways where you can test. Let's start big picture first. So there's a lot of tumor biopsy tests out there, and there's a lot of blood tests out there.
Yes. Yeah.
And I think the blood tests have really come on in the past couple of years, where we can take your blood and test your tumor. What are the big differences and advantages and disadvantages to taking a biopsy tissue and doing the sequencing of the tumor versus a blood sample of the patient and trying to do the sequencing of the tumor?
This whole expansion of technology and science and everything, we have these two major groups, like you said. You take a piece of tissue, and we call it a snapshot. So when you take a biopsy, that is just a snapshot. It's like taking a picture. You're capturing what you took out, and that's it. You're not getting anything that's surrounding, nothing that may be circulating the blood. If there's metastatic sites, you're not capturing that. You're solely capturing that portion of tumor that you took out. And that is really advantageous for multiple reasons, is that you are getting that full picture. You're able to send that for sequencing, and tissue biopsies right now are highly more sensitive. Since it's tissue that you can take and fully look at, you're able to determine the DNA, the RNA, and the protein, what's going on inside the cells and on the cell surface. And so it's a snapshot where you're able to capture heterogeneity and everything like that. Now, in contrast, liquid biopsies are very advantageous, because if a patient does have metastatic sites, essentially when you take a blood draw, you're taking a hodgepodge. If you're having metastatic sites that are essentially shedding tumor into the body, into the blood, you're able to take a draw of that. So I think of doing a recording versus taking a snapshot. So your chances are that you're going to capture cancer information from multiple metastatic sites. And really one of the pros of doing this is that it's less invasive. No one wants to have a biopsy, and sometimes, most of the time, they can't have an additional biopsy. So that's where liquid testing really comes in as a pro. However, there's a sensitivity issue. When you're able to take that snapshot or that picture and do a tissue biopsy, the material is right there. You can see the tumor material, you know you have it, and that can be analyzed. Whereas not all tumors shed, or mean that they spread and you can capture it in the blood. And so it's a little harder in sensitivity to be able to do that. But in the absence of tissue, at least you have a backup, which we use with liquid biopsies.
And when we talk about sensitivity, what does that mean practically? Does that mean we're more likely to catch something, we're more likely to miss something that's there? What does that mean in kind of layman's terms?
Yeah, so sensitivity is just what you said, is that we're highly sensitive in tissue, meaning like if the tissue is actually there, you see the tumor, you've captured it. Versus in a liquid biopsy, there's less sensitivity in the fact that you can't always detect it, because again, it's a hodgepodge. There's a lot of other things in that blood specimen outside of tumor. And so you're looking at your blood cells, your white blood cells, and everything else that could be found in blood. So you have to sift through things more. So if you have to sift through things more, your sensitivity kind of goes down, whereas in tissue, the tumor is blatantly right there, and you can assess it.
And I think one of the things that we're going to discuss in one of the later sessions about our circulating biomarkers is that like Peaches mentioned, not all cancers let their DNA float in the blood. And uveal melanoma is one of the ones where we know from really good data and really good studies that you're less likely to find uveal melanoma DNA in the blood in patients with uveal melanoma than you are other types of cancer like cutaneous melanoma or breast cancer. And so these liquid biopsies can be easier. They can give you a more holistic picture because you're looking at everything versus just one spot, but you are more likely to miss stuff that's there because of that. The other thing, just taking a step back, here when we're talking about the liquid biopsy, we're talking about that fishing expedition where we're doing sequencing to say we want to sequence everything in the tumor and figure out what changes does the tumor have. Our conversation in about an hour or two where we're looking at circulating DNA is really more of a focus panel that we're using to guide and measure, not to really understand the biology. So they are a little different tests. That's important in regards to the likelihood of finding something or not, because with liquid biopsies, the more things you look at, and with tumor testing, we want to look at everything for this discussion, the more likely you are to miss stuff. Where if you look at one thing, you're more likely to find that one thing. Talking about the tumor biopsies, you're less likely to miss any changes that are there. There are different types. There's targeted panels, I think some up to a couple hundred genes. There's some that sequence the entire genome. There's some that sequence both DNA and RNA. Can you kind of discuss the differences of the panels and the advantages to the different methods that are out there?
Doing DNA and RNA assessment, it's, as you can imagine, we have a lot of 23,000 plus genes in our body, lots and lots of genes. To be able to look at all of those, the technology has advanced quite a bit, and so that it's somewhat cost efficient for us to be able to do this. Now, when we look at IHCs, or basically proteins, what's on the cell surface of your tumors, that's very advantageous for us, but that's one kind of test. We call that, surface and IHC, a surface test. So it tells us what's on the actual outside of it. Can it be targeted? This is a lot of your PD-L1s and other proteins. And so a lot of your local labs, Sonora, Quest, and whatnot, they'll do those in-house. They can typically do that. But they don't have the technology to do full exome or genome sequencing. And that comes to a lot of your commercial labs that many of you may be familiar with. So when we're talking about whole exome sequencing, we're basically looking at analyzing DNA and RNA that are basically with whole genome sequencing. I'll just start there. That's a bigger picture. Yeah, it's a lot harder to do because you're looking at everything that's going to be made or not be made. Whereas whole exome sequencing, it's a lot more targeted.
So a lot of your DNA is what we call junk DNA.
Yeah.
It's there and does
it,
nothing.
That's what it is.
And so whole genome is you sequence everything, including the junk.
All the junk.
Where whole exome is saying we're just going to sequence the stuff that we know actually has a biological purpose.
Exactly.
Yeah, and that's the difference. And the importance of that goes back to what we discussed before. The more things you look at, the harder it is to know what's there. And so trying to focus on the non-junk we think is advantageous.
Exactly. And again with hotspot panels. So we always say 10 years ago there were maybe 50 or 100 genes that were actually relevant in cancer that we needed to know about. Then it was 300 genes. So when we started, we call them hotspot panels, where we just look at those 100 genes or those 300 genes because those are the ones most valuable in knowing about. But what we're seeing is that number keeps increasing. Now it's up to 700 and something genes that are relevant in cancer. And so we've moved out of this thought process of just doing what's currently relevant in cancer and expanding that by looking at whole exome, looking at everything essentially. And a lot of people say, well, that's a lot. Why would you be doing that? It's not even relevant right now. But then I can argue this gene wasn't relevant, 6 months ago, and now it is. And so the advantage of basically doing whole exome sequence, so not looking at just 700 genes, but looking at 20,000 genes, is that when you do that, you're basically getting that whole molecular blueprint of your tumor right now. And so if there is something that may be changed in the tumor, if there's a clinical trial or a therapy that may be upcoming, you don't have to retest for that tumor. You already know that tumor harbors that alteration. And so we always say that every gene is going to be a cancer gene at some point, so why don't we test for it now? Why don't we get that full picture of all the genes and their status. And then if you do another biopsy, you can also see how it changes later on. But at least you get a snapshot of what's happening, and not just a limited view of those 100 or 300 genes, but everything that's going on. So that's the difference between a hotspot, just looking at a couple hundred genes, versus full exome sequencing when you're looking at hundreds of genes at a time.
And I think what you said is important. In the future, every gene will probably be a cancer gene. Dr. Mamie, who's over there, and Dr. McKean will be talking soon about clinical trials. And we're seeing clinical trials come in with different ways of targeting vast number of mutations that are coming in the pipeline that will be available both now and in 2-3 years. And so having that information is hugely important. So I always tell people, if you're going to do genetic testing on the tumor, like I've already said, that's a fishing expedition. So I tell people, if you're going to fish, don't halfway fish, right? If you're going to do it, do it. And so it's really important, if you're going to do these tests, that you do these full panels, because most of the time you're not going to find anything that will affect how your cancer is treated or how your family member's cancer is treated, but you could, and we've all found something that has changed the management and benefited the patients.
And you also raise a good point. If you're going to get NGS testing, I feel like there's a lot of jargon in the field right now. NGS is used as a huge umbrella term for what we were talking about whole exome sequencing and whatnot. And so it's important. I always advocate people to know what they're looking for and having those conversations with your oncologist, as they're able to tell you what test they'll be sending for and what that test is actually going to give you the results for. You can ask them, how many genes are tested in my panel? And if they say 100, 200, you kind of know it's a hotspot. And maybe you can advocate for something larger. But of course, it's always up to you and the oncologist, but just knowing kind of what you're looking for and that there are differences in these tests, I think, are really important in your own journey.
And they're speaking to that as well, these platforms, this next generation sequencing or NGS testing is not cheap. No. Most of the time insurance covers it, and so insurance is paying a hefty price. But if you're gonna have your insurance pay thousands of dollars for the test, pay thousands of dollars for the complete test is what I always say. Make sure you're getting all the information if you're going to pay for it. And thankfully, within the past couple of years, most of these panels have become very comprehensive, pretty much complete genome or complete non-junk genome. But there still are some very focused panels out there. And so just making sure you send the most broad one you can find is important. So that's kind of a big overview of the different tests. What questions do you guys have before we start talking about uveal melanoma genetics and how this is impacting treatment in the next part? The question was, have we heard of the Gallery tests that are supposed to detect cancer, and do we think it could pick up melanoma or uveal melanoma? So that's a great question, and the Gallery test is a little different. So most of the time when we're talking about these genetic tests and these liquid biopsies that we're discussing, we're looking to say, are there changes in the DNA? So has the DNA changed its sequence or gotten shorter or gotten longer? This Gallery test actually looks at the methylation. So on your DNA, you have these little methyl groups, and then there's a different pattern of how those groups can be on there. And so that really looks at this methylation pattern and then can predict cancer. In the current version of the test, they can detect all cancers, and these were validated on very big trials. I don't know off the top of my head if uveal melanoma, how well that was represented in those trials. Usually rare cancers like this are not that well represented, and so the tests are not as good for rare tumors such as uveal melanoma as they are in more common cancers like lung, breast, prostate, etc. The other thing about those tests is they're still new and we're still learning. In the current version of the test, if it comes back saying that you have cancer in your blood for, let's say, for example, breast cancer, there's a 44% chance that you actually don't. That is a false positive. And so I think the technology is advancing. The newer versions of that test are in trial, so I think they will get better with time. Right now, I would be hesitant to put too much weight on it for uveal melanoma just because I don't know if it was adequately represented and how the test was developed. And a test is only as good as how well you trained it. Yeah. I don't know if you have any thoughts on that,
No,
Peaches.
I don't. I think it's exciting. It's, again, that whole expansion of liquid biopsies, and it's the new kid on the block, essentially, in testing. Again, the data is going to take a little while to catch up, so I'm always a strong advocate, like the NCCN guidelines on if you have tissue, that's what you want to send. But with all these additional tools with liquid, it's getting more and more exciting, especially with, you know, early detection and being able to determine if there's likely to be recurrence and everything. So we're in a really neat time of science and advancements.
So the question was, for people who are like 5 years out, are there any research looking at liquid biopsies to try to understand what's going on in the genetics of the tumor? And from my understanding, there's nothing in that particular area. Now, there's a lot of work on trying to develop blood-based biomarkers. So that can be circulating tumor cells, circulating hybrid cells, circulating DNA. And I believe we have a panel on that later this afternoon. Those are more focused panels or focused tests, really trying to ask the question of, can we detect the cancer in the blood so we can follow and monitor it to predict recurrence or response? That is, it's a similar technology, but it's a different application of it than saying we're going to try to sequence the entire tumor to try to understand what's driving the tumor and how we can treat it. And a lot of that goes back to how big of a scope you look at. You know, to really do the full sequencing, you need a good amount of DNA. So I don't know if there ever will be a situation where someone has no disease on their scan and we're able to do a somatic tumor test like Peaches is talking about, where we can get all that information. Just because if it's not on scans, there's probably not going to be enough DNA available to actually run the test.
Yeah, absolutely. That's very important, and we take it very seriously as a scientific community, is that there are a lot of different changes that can happen in your DNA, RNA, protein that actually aren't going to go on to have a functional effect. They're not going to cause cancer, and maybe we just don't know enough about that alteration right now. And so there's a classification of how we basically label if it's going to be cancerous or not, if it's going to contribute to cancer or not. And so, you know, when I talk about, oh, we sequenced 23,000 genes, all this information, that would be an encyclopedia, essentially. Way too much information. So I can speak to Karis at least, and a lot of this happens in the background with much of the bioinformatics is we look at the mutations, we see about the relevance in that specific tumor type, the relevance of that mutation in other tumors. Does it play a role in the cell growth, or in basically, is its function altered enough to cause cancer? And so we will classify them. We'll say, hey, this is a pathogenic mutation. This is something that's going to contribute to it. Or it's a likely pathogenic, meaning we see that it has, it contributes to cancer in lung cancer, but not so much in uveal. But it's good for you to be aware of it. And then we have other ones where there's mutations that we know nothing's going to happen with that gene. Essentially, you have that information there in case it does become relevant in the future. You could always give a call and get the information, but we know that it's nothing that you really need to pay attention to right now, because it's just, you know, a word that's basically misspelled, but it's not you can still understand the sentence, is how I think about it. And so, but it is very important, because you don't want anyone to be looking at a report and see all these mutations and get scared when they're actually not doing anything in the body. And so we do make sure to do that and classify these mutations so that we know that the ones that are relevant are actually the ones that are going to be provided to the clinician.
Point, Dr. Mamie. So, you know, when we're interpreting this data, there's a lot of questions we have to ask. I think the first question we have to ask is, does the change actually affect that protein, which Peaches discussed? And then there's a second question of, does that matter to this tumor? And that one's sometimes a harder question to answer. But, you know, like Dr. Mamie alluded to, we do have what we call driver mutations, which are mutations that we know are driving the cancer. And then we have what we call standby or bystander mutations, which happen in the cancer, but probably aren't driving it. And those can be sometimes near impossible to differentiate. But one of the things you can look at is the percentage within the tumor. So anytime you get these reports, you'll get a percent that says this change is in this percent. And sometimes we will see, you know, people being treated with drugs where Some mutations have 40, 50% in the tumor. They find one mutation that's 1 to 2% in the tumor, and we'll say, oh, we should try to treat that. Probably not a good treatment option if it's only in that few percentage of the tumor, because you're not actually covering it all. So you do have to interpret these reports and these changes, sometimes with a grain of salt, by asking the questions, does that change actually affect biology? And does that biology actually drive this tumor? Because sometimes you can get a little lost in the weeds in these reports if you don't look at those or don't ask those questions. So let's talk about what do we expect to find in kind of a testing of a uveal melanoma. So as most of us know, most uveal melanomas are driven by one of two mutations, either a mutation in GNAQ or GNA11. And so when we send these reports, we 96% of the time find a mutation in one of these two proteins. Those are proteins in right now where we can't actually target. There's no drugs that can actually turn that on or off. There's a lot of work going on in the scientific community trying to figure out how we can do that. But just the type of protein that these mutations are, it actually makes it really difficult to block. There's different types of proteins out there. Some are very easy to turn off and turn on. This particular type is a little bit more difficult, so it's been a little harder. Then a lot of the other mutations we see, or the other most common mutation we see in uveal melanoma is BAP1, which again is very hard, not necessarily a therapeutic option. So a lot of times when we do this type of testing, in patients with uveal melanoma where their cancer has spread, we see a mutation in one of those two, GNAQ or GNA11, maybe with a mutation in BAP1 or a couple other proteins. So most of the time, the fishing expedition of doing this testing doesn't necessarily change your treatment option. But it's really when we find something unexpected, because one of the big shifts that we see right now in the oncology community as a whole is redefining cancer not based on where it came, but from the genetic mutations. You know, 100 years ago when we started treating cancer, the only way to kind of lump cancer into groups is to say this is where it came from. That's why we call breast cancer, and prostate cancer. But we're learning now that the molecular signatures, for some cases, are actually more important than where it came from. So I have had patients with uveal melanoma that's had molecular signatures more consistent with types of colon cancer that can respond to immune therapy, and that will change what we do. And so most of the time when we're doing this testing, we're not going to find something, but if we find a mutation or a protein that is more similar to another cancer, that can actually change how we treat the patient and benefit the patient. So like I said, it's a fishing expedition. Most of the time you're going to come up negative, but it can actually change what we do and how your cancer is treated.
I feel like with the more information we get from all of these tests, we're going to learn more about the cancer itself. And so we know, GNAQ and all the other ones, which ones are prevalent. We can't act on them yet. There's clinical trials, but we're learning about what shouldn't be there, what's commonly not there, and all these signatures that we can potentially develop in the future to identify these or maybe find future targets. So that's a whole other research side of it. But, the more we test, the more we know, and hopefully you can advance it in the future.
I think the other side benefit I'll talk about is most of the tumor testing platforms that are out there right now will actually give you your HLA type, which is not the type, which is not the cancer per se, it's more you. HLA type is what I tell people is your blood type, but it's not the blood type we use for blood donation, it's the blood type we use for organ transplant. And for most tumors, it's not that relevant. For uveal melanoma, it's highly relevant. We know tebentafusp or Kimmtrak is only effective and approved for people who have a certain blood type, HLA A0201. And we know that there's clinical trials, such as the IDEA clinical trial, that's available right now for people who don't have that blood type. So it is advantageous to get this testing because you're getting that information as well, and you can really get kind of both of those questions answered in one test, which is great, where historically they were separate tests. Any other questions? No. Anything else you want to add or discuss, Peaches? I know the talk has been pretty informal since we didn't have slides.
Yeah. I always advocate for having the conversations with your clinicians. I know, Dr. Moser, you're fantastic with taking time with your patients, and I know, sometimes that's a little hard to get from clinicians. So, advocating for yourself, asking those questions about, again, somatic testing can mean a lot of things to a lot of different companies. So take that time with your clinician who should know essentially what you're looking for to get the right test.
So, Dr. Moser, Peaches, I have a question for you guys. Some of us, at least, are familiar with the Castle Biosciences report that comes in. We can see lots of different things. I know I've looked at one of my Caris reports with Dr. Moser in an appointment, but I'm curious, do you guys have any tools at Caris Life Sciences? Do you guys have any tools for helping patients to understand and interpret their report, aside from obviously talking to their oncologist?
It's a great question. So, we do. We have a whole patient navigation team, essentially, that will help. I like to work closely with my oncologists so that if they have questions, I can be that resource. I'm, in my role, technically not allowed to speak to patients in that capacity.
Yeah.
But so we do really good, we offer a lot of education things. So we work with RNs and the whole clinical staff if they want to learn how to interpret the reports as well, just at a basic level.
Yeah.
So that they can at least, what does the green box mean? What does the red box mean? Where am I looking on this report?
Yeah.
Because I think it's valuable. A lot of the time, they're spending time with the patient as well, answering these questions. So I would say a lot of companies do have some sort of patient navigation team that can help them walk through those reports, but we leave it to the clinician most of the time because we don't want anything to kind of get misinterpreted.
No, for sure. That totally makes sense.
Yeah.
Another question kind of just regarding what you guys do on your end. Obviously, biopsy testing, rare diseases, we know from many of us who've had Castle testing, and I know I've seen this on my Caris test. We get, an explanation of benefits, and your insurance comes back and says, this is an experimental test, and we don't cover it. What does Caris do for patients as far as a financial standpoint to help them figure that out?
Yeah. That's the two most common questions I get. No, that's an excellent question. So again, we have an entire patient team that will do that. One thing I can advocate with Caris is that I know the test is expensive, just like most tests. It really depends on insurance and where you're at with your deductible as well. That plays a huge part versus getting the test in the beginning of the year versus the end of the year, all of those logistics, which I've recently learned. But I do pride myself in the patient navigator team that we have. We don't direct bill or balance bill. Thank you. And so the patient isn't going to receive a bill like that. If they were, we've had it maybe happen by mistake once, you contact us immediately and we're going to work to figure that out. We also know that some have to do self-pay. And so how do we work with that? So basically, even if there were a bill in maybe a couple $100s, there's still a program for that.
Okay.
And so we're not going to dig into finances. We don't do any of that in the background, which I know some companies unfortunately do.
No, I think it's important to point out, especially for those of us who are dealing with, the financial aspect of things and just the confusion and the insurance battles and just all of the things that can come into our diagnosis. It's important to recognize we do have companies who are doing these tests who understand that and who are making it as much of a priority as possible for patients to not be financially burdened by having this test done. And I guess you guys can correct me if I'm wrong, but my understanding of it is that these companies want to do the research. They want to see what's happening, and it's worth doing the tests, and it's worth seeing the results from as many patients as possible to get that information. Like I don't know, I guess for me as a patient, it kind of feels like the information feels more valuable than the cash flow, if that makes some level of sense. Like, I know the cash flow is important. The company has to continue, but I just appreciate that Caris, Castle, that they make a priority of trying not to put that burden on patients.
Yeah, and both of the companies, we work closely with Castle too. They're great. It's how much more can we learn about this so that we can have advancements. It's a huge, so translational. So that's the whole goal.
Yeah.
Is that we learn all of this information, find those. So I would even say that the drugs that we have now for these specific markers, like the HLA or PD-L1s and stuff, that all came from learning about it first, so it being in the experiment, and now you went through clinical trials to now be a drug that's available. So it all begins with research, and with Caris, we're twofold. So first and foremost, it's about getting those reports to our clinician to help with treatment. But our second branch is research. So how do we use this, not to sell the information, but to learn about it, and then partner to figure out, like, hey, these genes are coming up a lot in this cancer. Based off of how they work, can a drug be developed to then, go into clinical trials for use?
So just to recap that question, for those of you who didn't hear the question, it was just, does the more testing that happens make it so that the drug becomes approved as something that is no longer experimental by insurance anyway? Kind of to go along, go ahead.
I'll answer that too. So you're absolutely right? If we find that something's there on doing these tests, that drives the development of ways to try to target it. And so by doing more tests, we might better understand it. I think the other thing too is, when these tests are done, the companies can share the data kind of anonymously in aggregate. So they'll say, here is the data of so many patients with this that's done. And we are starting to do that, and through academic partnerships, I've been involved with this with Caris and other companies. We're going back and saying, okay, so you do all the RNA, and that's not part of the report, but can we try to analyze that to better understand the tumor biology? So both, getting the test done helps potentially drive new therapies by finding stuff to target, but we are also exploring that in an anonymized way or non-patient specific way to try to understand the biology so we can better treat it in that way too.
Yeah. Awesome. Do we have any other questions from the audience? Dr. Memi?
Yeah, I was going to just further Dr. Moser's point that, when we collect these NGS or DNA sequencing or RNA sequencing data, we can pair it with the outcomes that have happened, patients, mutations, and how they did with certain treatments and what kind of side effects they had. And hopefully that could drive clinical trials and research to predict responses or predict toxicity in patients. So, if we could say that, hey, there's a marker Z in this patient's tumor, and that predicts for toxicity, but it doesn't predict for response, then we can potentially avoid those therapies, right? And so that will only come if we have paired data from the patient and from our industry partners, Caris, Tempus, and all of them, and putting that together in an anonymous way, so we're not really, sort of red taped by a lot of the things that happens in this kind of research.
Thank you so much, Dr. Memi. I think one of the things that I take away from this is these kinds of tests help eliminate trial and error. Like right now, I remember when Dr. Moser came out to, with Meredith McKean in Nashville, one of the things that we talked about at the very beginning of some of those sessions, we were talking about, navigating clinical trials. We were talking about the new FDA approval of tebentafusp and Kimtrak. I guess that's the same thing, in case you didn't know. But one of the things that Dr. McKean and Dr. Moser both touched on is that at that time, 2 years ago, most uveal melanoma oncologists were kind of like flying by the seat of their pants. It was guesswork. We're getting closer and closer to having less guesswork for certain parts of the demographic of patients. And the more of these kinds of tests that happen, the more that guesswork goes away, and it's no longer trial and error quite as scary feeling as it, was 5-10 years ago. Okay. Any other questions, comments? Let's give a round of applause to Dr. Ulrich and Dr. Moser, and we'll bring them back up a little later.